Are you able to see the screen now?
Yeah, we can see him.
Oh, great.
We can see you.
Good morning, everyone. And it's an honor to present at this conference alongside great. Looks like I'm having some difficulty. Oh, come on. Sorry, there are some difficulties in my machine. Okay, so it's
Okay, we can see your. Okay, we can
honor
see
to
your
present
voice.
over here alongside great people like Professor Harkam or Professor Tino and others.
You have to share the slide.
Oh, are you not able to see my? Come on, I'm so sorry. Okay. Sorry, actually the problem is because of the two screens, and that's where I'm facing this issue. Yeah, my desktop.
It is good now.
Sorry. Okay. How about now?
Yes, good.
Okay. I'll just take this on the other screen. Okay. Without delay, I'm going to talk about the data curation today, which is kind of a forgotten practice in the era of artificial intelligence and machine learning. Everybody is talking about artificial intelligence and machine learning nowadays, that they have been building models for QSAR or all of the complicated endpoints, but nobody talks about the data curation. And hence, I have been working on building a lot of these models as well, but I just want to share my experiences of what kind of data pre-processing is required when you're building these QSAR or QSPR models. Just to give you an overview of my company, actually, we are a software and a consulting company where we integrate machine learning, pharmacokinetic simulations, and systems biology with many more complicated and complicated processes. This talk is focused only on my efforts related to data curation and pre-processing, as well as data validation, while building the models for predictive QSAR models, predictive QSAR and QSPR models in ADMET predictor. Just to give you an overview, actually the validation is absolutely necessary, whether you are talking about news or activity data. Here are two examples of long-lasting fake news. First one is a photo published in Daily Mail, which has been well known as the London surgeon's photo of a monster, which is a Loch Ness monster. This was published in 1934, and other one was a Bigfoot, which was sighted in California, published in 1967. Since then, thousands of people have visited Loch Ness West as well as California, searching for these monsters with no luck. These news become kind of real news in the real life or as a part of regular life until someone with higher authority or with a power calls them or proves them fake. Until then, people still believe that these news are the real news and go on following those actually. So we will revisit these pictures once again in few minutes. Meanwhile, I'll just like to continue with our data curation talk. Before another picture which I like to show is maybe many of us or many of you might have seen these. Recently, due to COVID-19, there have been a lot of this news, specifically this picture, has been circulating in social media. The fact check of this picture revealed that this picture was originally published in 2014, where people lay down in Germany in remembrance of 528 victims of catchback. So this picture is not related at all to COVID-19, but people never bothered to check if this is the correct if this is the right picture or not. They just wanted to share it, and people started believing it that COVID-19 resulted in this kind of deaths. Let me show my disclaimer before I dive into the data curation part. This is very simple. I have no intentions to evaluate or criticize any public or commercial databases, and I don't want to hunt for the errors in existing research articles. What I will be sharing here will be my experiences while building these models. So my overall intention is just to suggest how we can utilize these existing databases efficiently, as well as if I can provide you some strategy for database curation, chemical data curation, that will be an additional plus point. So why data curation is necessary? This is the email my colleague received recently, few months back, from our client or customer, and they were evaluating our software, ADMET Predictor. And what I would like to highlight here is we have done some test data sets through ADMET Predictor, and we get rather poor correlations. This is what actually was really disturbing to us. So we requested them that if they can provide us the data, and they did actually. So they had provided this plot to us, telling that this is what the actual versus predicted human liver microsomal clearance model. And this is the performance of our clearance model against the experimental data. And they also provided that the data was collected from Campbell database. When we downloaded the data and we did some data curations or some conversions, we did not, it was simple conversions and curations based on the manual as well as common sense actually. We see a very good correlation between our prediction and the experimental data. So what did these investigators miss? Actually, this is the plot what they have sent us. But what happened is when they carried out the comparison, they missed on the part that ADMET Predictor provides the HLM clearance that is corrected for microsomal binding. Those who are working in pharmacokinetic modeling or ADME modeling, they know that microsomal binding is the correction of microsomal binding results in unbound clearance. So the prediction that ADMET Predictor provided was unbound clearance, not the regular bound clearance. So when we converted this into a into the unbound clearance, we started to see some correlation over here, which is not great. But the other part, which investigators didn't bother about or did not pay attention to, where there were 84 compounds in this case, where the clearance value was clearly reported as greater than 150, or there were around 270 compounds where clearance value was reported as less than 3. That means these were the limits of quantification. Now these values are not the specific endpoint values, and we cannot use these values when you're comparing the prediction from the software. So if you get rid of these 350 compounds, which are highlighted in black over here, now we start getting to see a very good correlation between predicted as well as between the experimental and the predicted clearance values. So this is why actually I have been mentioning that the data curation seems to be an absolute necessity. Most of the people in the field think that the model building, QSAR model building, is similar to what they had done, and just follow or download the data from literature or download the data from chemical data sets and use that data as is in building the models. Yesterday, I think Jagmohan mentioned that garbage in, garbage out, and that strategy that works for each and every model, whether it is a QSAR model, whether it is a molecular dynamics model, or whether it is you are carrying out molecular docking. You have to be absolutely sure that whatever data you are providing to computer will. You have to make sure that whatever data you are providing to the computer is of high quality, and then only you will get a good model of high quality. So this is the outline of my talk. I'll just discuss what is data validation, where do these errors come from, and how do we find them. And at the end, I will give you some more examples. Why should we care about it? I have already mentioned one, but I have few more examples why actually it is absolutely necessary to care about the data curation. So if we look at the Oxford Dictionary, it says validation is an action of checking or proving the validity or accuracy of something. And in terms, when you talk about validation in computer science, the data validation is a process of ensuring that the data have undergone data cleansing to make sure that they are both correct and useful. Without data validation, the data, whatever you get, is of no use, whether you are talking about computer science data or drug discovery data. And validation in drug discovery, specifically early drug discovery or late drug discovery, is an absolute necessity. The research has shown that on an average, there are 2 papers per medchem publication, there are 2 errors per medchem publication, and this number or this error rate can be as high as 8%. And apart from these errors, so where do these errors come from? These could be experimental errors, or these could be errors due to reporting or during the publications or printing. But apart from that, actually, once the publication is out, the data extraction and digitalization by the database extractors, they introduce another set of errors on the top of the errors in the publication. And that's why actually whenever you are extracting the data from databases, you have to be extra cautious about using that data directly as is. And for accurate and predictive models, you have to make sure that the clean and accurate data is provided to build these models. Unless your model will be just another extra model in the published domain. Dozens of databases are available out there. Some of them are public databases, some of them are commercial databases. So Campbell, Binding DB, or PubChem, they constitute the public databases, while there are others which are public commercial databases. The usefulness of public data sources is questionable due to lack of the necessary quality control. Hence, many people and a lot of big pharma, they prefer commercial databases over public ones because there is someone they can hold accountable for. I do not agree with this. I will show you my own reasons why I believe commercial databases are equally well, there is no difference between public and commercial databases as long as we are looking at the quality, as far as we are looking at the quality. But once we get the data from these databases, unless we wash them or rinse them, and sometimes you have to scrub them to be useful for building these models. The usefulness of this data and the data curation, which I will be talking about. I'm not the first person to show the significance or the importance of these processes. Several well-known research groups have already set up the base for this talk. All I will be doing here is I'll just stand on their shoulder to see what others have missed. And if we look at these few landmark papers in data curation literature, you will see that most of these papers are talking about chemical structure curation for the QSAR or chemical data curation. And going through these papers, they are mostly talking about, okay, you have to standardize the paper, you have to standardize compounds, you have to make sure that you're using right tautomers. Yesterday, Devin talked about tautomers. Neutralization is necessary, make sure that the stereochemistry is absolutely right, you are not using mixtures, and so on. When we are talking about QSAR models or QSPR models, apart from chemical structures, quality of biological data is absolutely necessary. And there are papers talking about these as well, where they are comparing commercial and public bioactivity databases or finding out the error rates in bioactivity data. So I will be discussing further how we can find these errors, or what are the reasons for these errors, and how we can find these errors. And that's where actually I will reach out to my another point. Where do these errors come from? There are multiple sources of errors. Let's start with the easier one, data extraction. That means blame the others. The data extractors, whoever is extracting the data from publications and putting them in the databases, let's blame them first. And then I will discuss where we stand over here. So whenever the database extractors are extracting the data, there is a possibility that sometimes they do miss putting appropriate units. So for example, if we look at the terphenadine bioactive HERG IC50, let me rephrase it. If you look at HERG IC50 of terphenadine for two different records, we can see one record show that it is 56,000 nanomolar, and the other one record shows it has 56 nanomolar. So how did this happen? So what happens is when database curator extracted the data, they extracted the significant digits correctly, 56 nanomolar, but at the same time, for both the papers, it has been reported as 56 nanomolar. Unfortunately, when the database extractor pulled out the data, instead of writing 56 nanomolar, they reported it as 56 micromolar, and that's where the inconsistency arises from. Another example of data extraction is providing the wrong parameter, not the wrong units, but the parameter. So if I tell you that the bioavailability of decipermine is 15%, would you believe that? I think you should. Why? Because the range for fraction bioavailability is 0 to 1, or let's put it 0% to 100%. So 0.15 or 15% bioavailability is within the range, and it looks realistic, actually, quite realistic, until if you see the enzyme cell assay description, where it mentions that this number, the 15%, represents fraction of compound unbound in human plasma. That means this number represents the plasma protein binding value and not the bioavailability. And fortunately or unfortunately, the range for fraction unbound is the same, 0 to 1. So how do we find them? Most of the times, these kind of compounds, when you are building the models, these kind of errors do stand out as outliers. And we have been trained since our school days, or since our graduate schools or master's days, that okay, if there is an outlier, we should not include that molecule in our modeling data set. We should ignore that molecule, and that's where actually the complication starts. So we have to be very alert when we are using the data. We have to make sure that the parameter details or the activity details, enzyme cell assays, we have to be very sure to check each and every aspect of that data. Next source of error is endpoint confusion. This is the data set which I was creating during while collecting the data set for classification model for breast cancer resistance protein inhibitors. In this case, actually, we collected data from public as well as commercial databases. The commercial database search returned two compounds. They were having very high potency and low IC50 values. That means they were very potent BCRP inhibitors. But when we looked at these compounds, all these 44 plus 23 and these 39, all of them were positive compounds. So that looked a little suspicious. When we go back to original papers and look at the assay description, that suggests that these IC50s were reported as cellular IC50s or not the transporter IC50s. That means these are the cytotoxicity data. This is the cytotoxicity data and not the IC50 against BCRP inhibitor. Now, what happens is this, if we had included these compounds in our data set, we are talking about around 100 compounds out of 700 inhibitors. So that constitutes 15 to 20% of the data set, all of them positive. So if we include these in model building, that will influence the model dramatically. So we have to be very alert while using, we have to be very careful while using these kind of data. Next source of error is original research articles. We are still blaming others at this moment. We are still saying that, okay, we are taking care of everything. But in this case, when I say original research articles, sometimes authors do make mistakes. For example, same set of authors, same research organizations published a single molecule in two different papers having hERG IC50, in one case is showing that it is 18 micromolar, and in other case it just shows that it is 18 nanomolar. So this kind of error, well, here database extractor did not make any mistake. Rather, the mistake came from the authors, but that doesn't mean that we should ignore that. Our model, when we were building the model for hERG IC50, we removed this compound and carried out the prediction. We found that this molecule is highly potent molecule, suggesting that it has a nanomolar activity and not the micromolar. Next source of error is database users. That means us who is using the database. How we are contributing to these errors is something is very unique. Most of the times, the programs where we load these data, they are equipped in such a way that they strip the extra salts or the counter ions from the molecules, and what we see at the end is simple molecule, not entire salt. But what happens is if you ignore the salt or the counter ions, you cannot compare the activities or toxicities of different salts altogether. This data was downloaded from Environmental Protection Agency, a government agency data set, and to build a model for rebuild dose or toxicity, to build the toxicity model for compounds. And initially we were surprised to see that the same molecule in the same data set, in single data set, have different numbers until we found out that they were talking about different salts. So we have to be very careful when we are comparing the activities or toxicities of different salts. It is absolutely necessary that we should treat them separately or differently. And the last source of error, which is one of my favorite actually, is we call it ABCD, that is automated and blind compilation of data. Nowadays, actually, the database compilation is carried out mostly by computers, computer programs, and very few human efforts have been put in while doing the database creation. So it is tempting to automate the curation itself by accepting as correct, but potential for false positive reinforcement is dangerously high. So what I want to show here is this compound 21, which we came across while building the model for rat plasma protein binding. So if we see the structure over here, this suggests that we have an exocyclic double bond on this ring, and this ring was transcribed the exactly same way in the Campbell database. When we started building the model, this molecule always jumped out as an outlier in our model, in-house model. Even if we include it as a training set or a test set, it was always predicted to be far away from its actual plasma protein binding, experimental plasma protein binding value. So we decided to look at this chemical structure, and what we see was actually shocking. In this case, what we see is this is the snapshot from the paper. And if we zoom this in, what we see here is number 21 was actually shown the way it has been drawn, what I have drawn in here. But if you look at this is a GMAT chem paper actually, and most of the GMAT chem papers have IUPAC name of all the chemicals. So if you go back to the original IUPAC name of this compound, that time we realized that the oxygen was missing. Because of this table or smaller row, or we could not, the program which extracted this data could not see this oxygen, and it just transcribed it as exocyclic double bond. So when we updated the structure and put it back in the database, in the model building data, we realized, okay, now it worked out pretty well and it was predicted perfectly on spot. So these are some of the errors actually, which some of the sources of errors which have been ignored or which are overlooked by many of us when we are building the models. So, but how do we find them? Actually, this is a very good question, and many people ask me how can we find these errors? Actually, we are talking about single molecule or a smaller group of errors when we are building the models using hundreds or thousands of data points. Easier way would be look at activity cliffs or toxicity cliffs. So this is another endpoint, and this is another series which I came across while building the LD50 model again. So there are 31 compounds having similar chemical structures, and LD50 value of these 31 compounds ranged from 0.25 to 77 mg per kg. Except the compound which is shown over here, zoom in here, this compound had LD50 of 3955, which is almost 4,000 milligrams per kilogram, which was very unusual when we talk about different, very close analogs of these compounds. So nobody had bothered to check why this is so, and in the competition where we built these models, nobody had pointed out these errors. Maybe the reason was this data comes from the paper, which is very old, I would say almost 35 to 40-year-old paper. So getting this paper was very difficult. But once we found this paper, we realized that the compound which I showed you is over here, compound 89. And when we converted this micromolar data to mg per kg, we see that it is 0.322 mg per kg. On contrast, the reported value is 3995. That means the database extractor who had extracted this data, he missed to put one decimal point, and that messed up everything in the dataset. Similar example was with this site and this kind of scaffold. Unfortunately, we could not get the access to this paper or this report, and hence in this case, what we had done, well, just to give you an example, give you an idea, LD50 for this molecule was reported as 7,000 liters per kilogram, that means 7 grams per kilogram. But 11 most similar structures for these compounds had LD50 in the range of 1.6 to 15. So we decided to remove the entire chemical series from building the models, and we predicted the LD50 for all of them. And our analysis suggested that the expected LD50 for this compound is in the range of 6 to 12 milligrams per kilogram. Just to remind you, we did not include any of these compounds in building the model. Yet our model suggested that LD50 should be somewhere like 7, not the 7,000. Another approach how we can find these errors is look at molecular match pair analysis, and that gives you an easily idea whether something is wrong with one compound or the entire data set. For example, if we see this, I just use molecular match pair analysis to find out if there is any specific, why there is so much of a difference between these pairs. The only difference between these two compounds is carbon to nitrogen replacement. Similar replacements in other molecules, for example, over here, the carbon to nitrogen replacements, they did not affect or they did not change the plasma protein binding very significantly. There are very minor changes or minuscule changes in plasma protein binding, even though people have changed the different ring systems from carbon to nitrogen. The only pair which stood out over here was this pair where there was 51% plasma protein binding difference of 51% in these two compounds. That's how actually we can easily find out whether there is anything wrong with the data that we have downloaded. And even if you are looking at these pairs, you can immediately find out those singletons or those few set of compounds which could be of problematic data when you are building the model. Another approach which I described in last slide was leave group out. That means whenever we see some one or two compounds which are outliers, we just remove the entire group of those group of those compounds and build the model, and then carry out the predictions of that set of compounds. So this is similar to people have been using leave one out approach. So here we leave the entire group of compounds, build the model, and run the predictions for the group which was not included, and see if the prediction, if there is anything wrong with those. So for example, in this case, while building the model for rat liver microsomal clearance, we found that these set of compounds were reported incorrectly in Campbell dataset. The reported units for these compounds was microliter per minute per milligram. But if we look at the original paper, the actual units from the paper were milliliters per minute per gram of liver. And those who are working in pharmacokinetic simulation, pharmacokinetic data modeling or ADME modeling, they would realize that this liver to milligram of protein conversion is a 50-fold conversion, and it needs a special scaling factor. Another example is over here. In this case, actually, we found that there was unit discrepancy of 1,000-fold. We excluded this set of compounds, built the model, and carried out the predictions. These compounds were predicted to have 3,000-fold activity than what they were reported in the database. We also confirmed these numbers from the paper. That's how actually the leave group out is a kind of a savior for us when we are talking about the entire group of compounds from one paper. And we have to be very alert about the numbers. I know we have been told that whenever you are building the models for QSAR or QSPR, wider range is always a good is your friend, let me put it this way. But that doesn't mean that you should be looking at PIC50 of 36 and including that molecule in the data set. PIC50 of 36 represents, well, 12 represents a picomolar molecule. Now think about it, if we are talking about PIC50 of 36, what kind of decimal points we are looking at. But the database extractor here did mess up. Like for example, the hERG IC50 for this compound in the paper is reported as 36 and that too micromole. Unfortunately, the database curator just reported it as PIC50 and that we just realized that while building the model that this molecule stood out by itself, having 36, PIC50 of 36. So we have to be very careful that yes, this is very high number, but will you include it in the data set? You have to be very wise enough to decide whether yes, you can include this. And this is another example. Actually, we have to be alert about the activity data. So when I was building a model for ratio of blood to plasma, and I realized that after building a crude model, this was one of the intermediate models. I realized that there were 35 compounds having blood to plasma ratio, experimental blood to plasma ratio of 1. And there were 22 compounds having blood to plasma ratio, experimental data of 0.55. This does not sound normal because if you look at the chemical structures of all these 35 compounds, they are very diverse groups. Same thing happens with the 0.55 data as well. From naloxone to raloxifene, these are, I would say, quite diverse data and diverse compounds, but they still have the same blood to plasma ratio. So and these data sets, actually, this was a data set which I compiled from the literature and has been used by various researchers in the past as is. They never bothered to take care of, to have a look at these set of compounds. If you go back to original papers or original references of these compounds, now you will find, let me show you over here one by one, actually. They had specifically mentioned in all these papers that if the data is not available, RBP is assumed. For acidic compounds, it is 0.55, and for basic compounds, it is assumed to be 1. For example, for naloxone, it is assumed for naloxone to be 1. Similarly, if you look at raloxifene, they also assume the RB value to be 1 for basic drugs and for 0.55 for acidic drugs. And eventually, this is another funny part, some people report it as 55 and yet mention that because the others, that means the Cubitt et al., this group assumed it to be 1 or 0.55, we are using it 0.55. So this is something which we have to be very alert about when we are building the model. We cannot simply use the assumed or the data which looks suspicious. What happened in this case is when the database curator extracted the data from here, they just noticed the numbers in the table and ignored the fact that these numbers have some footnotes and we have to be taken care of. These are some. Next is the user experience. We cannot just rely on various tools or techniques which are used in Cheminformatics. For example, I showed you activity plus or leave group out or match pair analysis. User experience equally counts, equally important when you are doing the when you are doing the data curation. For example, this example was provided to me by my colleague Bob Clark. He was building a model for ester hydrolysis, the metabolism site of metabolism model. And while doing this, actually, he extracted the data from World Drug Index, and there were few compounds. For example, the one which is shown over here is roxabolone, and roxabolone is present as two different um, prodrugs. One is dodecyl, and the other one is decyl roxabolone. When he built the model and tested his model for predictivity, he realized that in one case, the output was entire drug molecule and the side chain with even number of carbon. In the other case, it was odd carbon long chain. So it rang a bell in his head because of his experience, he knew that mammals have problems in metabolizing the odd carbon long chain, and we never have those kind of fatty acid chains in the products. So he immediately looked at the original structures from the WDI, and he found that in one case, actually this is easier. Yeah, in one case we have ester, and in the other case they have reported it as a reverse ester. Had it been the case that I was building this model, I would have never caught this error because I wasn't aware until he told us that mammals have problems in metabolizing the odd carbon. So this is what I was mentioning. The user experience equally counts when you are doing the data distribution. So far, I have shown where we get these errors from and how do we find them. Now let's see why should we care about them. Let's visit our first slide, which I showed you in the very first slide where I showed you the surgeon's photo and the big. There is a saying, they say, a lie repeated 1,000 times became a truth. If we, these news were believed to be the truth for a long time. So we are talking about 6 decades. In 1993, Duke Wetherell admitted that they had made the monster out of some plastic and clockwork and toy submarine. So the picture, what they had shown in here, was a fake picture. It was a fake news. Similarly, there was this Bigfoot news. Ray Wallace, this is the statement from their family. Their family revealed a secret that Ray Wallace had created the hoax. He made an oversized footprint with a set of carved wooden feet, and then he just showed them those footmarks, showing that the Bigfoot exists. Even after these news or this was revealed that these news were fake, let me show you something here. These are 2017 news headlines. And even in 2017, this was revealed in 1993. And even after 24 years or 25 years, 2017 has been a record year for sightings of Loch Ness Monster. And similarly, this is again 2017 news saying that Bigfoot sightings reported in California. So people are still believing that these monsters do exist. So this is I'm talking in terms of the news, but this happens in chemistry or in drug discovery as well. I'm recently building the models for site of metabolism, prediction of site of metabolism. And the model there was a model published in 2012 by Zeretsky et al. It was known as RS Predictor. They built a model for radioselectivity of 9 CYP isozymes to predict the site of metabolism. And they generated a data set of 680 compounds with all these 9 CYP isozyme sites of metabolism and provided it or made it available to public in supporting information of this paper. This data set has been used widely by many research groups to build. Everybody is trying to improve the site of metabolism model. So they have been building these models to improve the site of metabolism. And this is the latest paper which I came across. They have been saying that, okay, Zeretsky data set has been successfully applied in several other studies and is well established in the field. So if we look at these papers, they have been published since 2012, 2013, there have been published papers in 2015 and 17 as well. And recently, Finkelman and his group used the same set of molecules based on and used ab initio calculations for sites of matter. Now, when we looking at this, looks like the database, entire database, seems to be a very reasonable and accurate. We started to decide, we decided to check the authenticity or correctness of these structures. And what we find is there were more than 25 chemical compounds that needed a correction. For example, there were few compounds for which carbonyl oxygen was. There were few compounds for which we see a altogether different connectivity. There were different ring sizes or different positions of nitrogen. And there were few places where the entire side chains were missing. If we are only talking about one or two differences, that is still considered okay when we are talking about such a large data set. However, think about it like somebody is using ab initio calculations on the wrong structures and running the sites of metabolism prediction models and providing improved models what they have been done. Before it becomes absolute necessity that before running even the ab initio calculations, we know ab initio calculations are very expensive computationally. So before running the ab initio calculations, doesn't it make sense to check the chemical structures or authenticity of these chemical structures? And this is something I was talking about, that this lie or a lie repeated 1,000 times becomes the truth. Same thing happened in this case actually. Somebody has created the data set and people started using it. Nowadays, nobody even bothers to check the authenticity of that data or the correctness of that data. And this is something which is actually a serious or dangerous in terms when it comes to the molecular modeling and building the correct model. This is another example, actually, amiodarone and amiodarone hydrochloride. Both these compounds, this is a snapshot from PubChem for both these compounds. So PubChem reports that the solubility of amiodarone is 700 milligrams per liter, and solubility of amiodarone hydrochloride is 700 milligrams per liter. Anybody who is working in solubility area or who has knowledge of physical organic chemistry, he would disagree with that, saying that, okay, if it is a hydrochloride salt, it has to have higher solubility compared to the naked or nascent one. When we were building a model for solubility in ADMET modeler, amiodarone always stood out. We always found that it was an outlier. So the nominal solubility of 0.7 mg per milliliter or 700 mg per liter is predicted to be much less by our ADMET predictor. And down the line, if somebody collects the data from PubChem and starts using this kind of data, think about the quality of the model, what you will be getting from these data sets. This is another funny example. So while doing the literature search, my colleague Marvin Waldman came across this pentazine compound. And to show you what this compound represents, this compound is a hypothetical compound that consists of 6-membered aromatic ring containing 5 nitrogen atoms. Now that means this has been it has been already reported that it is a hypothetical compound. But if we see this, here are my points. This is a non-existent compound. It got into literature for computational work, trying to rationalize that it does not exist. But yet, this compound has a CAS number. And surprisingly, this compound is available from various chemical vendors. So my point is actually think about the company who is ready to provide you a compound which is hypothetical compound. So once this happens, it becomes virtually real compound. Why am I showing you this? So most of the times when we are doing the bioisosteric replacement, this is a very well-known example of bioisosteric replacement where phenyl group is replaced by pyridyl group, diazepam and bromazepam. Down the line, somebody who is not aware of pentazine and its authenticity, I'm pretty sure somebody will come up with a compound which is designed with the pentazine analog. And I do see it. I do not see that it is not possible. Actually, it is quite possible somebody could come up with that. So it is very important when you are looking at the compounds or when you are getting the data from the literature, please check the authenticity of the data before you use it for model building or any other computational exercise. Saying that, I already mentioned that validation is necessary, and I have shown you that these pictures have been the fake pictures, these news have been the fake news. That doesn't mean that the black swans don't exist. So this is an example that silicon is a group of fish that was believed to have died around 90 million years ago. They're abundant in fossil records, but it was not available. Until 1938, a fisherman caught the first living silicon. So yes, it is possible that sometimes whenever you are looking at some data, I'm not saying everything is rotten data. You have to be very careful when what to trust and what not to trust. If you can find the authenticity of data, there is no reason we should exclude that data. So this is an example which I show that yes, there do exist black swans in the activity cliques as well. For example, these are the three compounds where we see the PIC 50 value against the hERG data is ranging from 4 to 6.8. This is a wide range, and the only difference what we see here is acid to ester or the tetrazole range. But these kind of activity clips do exist. All we have to do is validate or verify what we see is real and not an artifact of a human model. And then we can include them in the data set. While doing this, many people ask me, can we automate this data? I would say we can. You have to be very careful while doing that. This is a paper published when somebody used 9 to do the automated curation, and what they missed over here is most of the times they used inchy keys, and what they did is they converted these kind of compounds in the scaffold, which is very different than the original set of compounds. Similarly, these are the salts that they had created an actual bond between sulfur and iodine, or similarly, the acid structures were converted into enols. And this is not only one compound, actually. There are a lot of compounds with these scaffolds or this scaffolds. And there are 155 acids which were converted in these acids. So you have to be very careful when you are using the automation. I'm not against the automation, but it is completely the user based and user dependent process, and user is completely responsible. And this is a saying what we all know, that one rotten apple spoils the rest. So if you are using the molecule, one wrong molecule in the data set, or one group of molecules which is wrongly annotated, I'm pretty sure your model is going to be equally rotten as well. So we have to be very careful while using or dealing with these rotten apples. And secondly, it is completely up to us to validate or not to validate. If you build a model with a regular data or without taking extra precautions, you will just build another model. There are hundreds of models being built and published every day. You will be just one of them. But if you take extra precautions and use the good and correct data, I'm pretty sure your model will be an extraordinary model compared to extra and another ordinary. We have been told, almost at the end, we have been told that whenever there are some compounds which are outliers when you are building a model, we ignore those. So it is easy to ignore the problems, but it is very difficult to face that problem and solve it. It gives a great satisfaction. I have been reviewing a lot of papers. This is I had reviewed one paper asking about why did they exclude some compounds. So this is what my question was, that, okay, only 42 compounds were used for model building, and another model was built using 51 compounds. The author replies that, okay, we omitted 9 molecules due to decreasing predictive power of these models. And as I said, ignoring is very easy actually. But what is important is find out why these 9 molecules have different or decreasing predictive power, and can you bring them up to the mark of 51 instead of just building a model of 42. Finally, this is the conclusion which I have discussed till now. The poor quality data is actually enemy of effective application of machine learning models. We have to be very vigilant while using any bioactivity databases or compilations. And of course, watch out for these ABCDs. One important part which I'd like to mention, solve the problem instead of ignoring it. And lastly, if you see anything, mention it to others. Tell them or spread the word that this is wrong or there is something wrong with this data set or this database. And that way, actually, somebody who had made that mistake, they may go back and correct their database for future generations to use it accurately. There have been a lot of people involved in this all whatever models I have been building on. This is my team at Simulations Plus, and thank you all for listening. I know I'm almost at the hour level actually. So if you have any questions, I'll be happy to answer them. Hello?
Yes, Pankaj. Thank you for a nice presentation. And yes, yesterday also, Dr. Ramesh was talking about this aspect. But Ramesh Shukla was saying that how we should be careful with the data. Well, but many a times this data could